What's up, TUMNAD lectures? In this video, we are looking on what we call the ECG. If you find this video helpful at the end of it all, please make sure you like the video, comment down in the comment section, and please subscribe. Let's go ahead and get started. So now in this video, we are going to be looking on the detailed part of what we call the ECG. In the previous video, we looked on what we call the basic components of what we call of the normal ECG, but we did explain how those kind of the patterns, how they were coming about. In this video, we are going to go into an extra mile and explain how all those kind of the waves on a normal ECG, how they come about. But before we go into the detail of that, we need to first understand some of the basic terminologies during the study of what we call the ECG. If we are to master the interpretation of the ECG, we need to first know some of these common terminologies. What are these terminologies? We need to first understand what is depolarization. We need to understand what is re-polarization. We need to first understand what is a lead. We need to first understand what is electrode, and we also need to understand what is a vector. Okay, those are the basic information that we need to first understand before we go very far into the detailed part of what we call the ECG. In this video, we are going to be looking on what we call the depolarization of the contractile cell of what we call of the myocardium. Now, we have extracted only one cell on the contractile myocardium. We want to see how this cell is going to be getting depolarized. That's going to be the main reason in today's video. Let's go ahead and get started. So now this one is going to be a cell. It's going to be part of what we call a contractile cell. And of course, there is also another cell which is going to be starting from here, going the other side. We want to see how this kind of the depolarization from this cell is also going to be leading to the depolarization of the next cell. Because that's how depolarization of one cell leads to the depolarization of another cell through some kind of the proteins, which is we'll talk about, which is going to be allowing the movement of different kind of cations. Okay. Now this cell, let us first pretend that this cell is at rest. And whenever a cell is at rest, like for example, this cell is going to be electronegative inside this cell. Now it means this cell is going to be having what we call resting membrane potential. Okay, we call resting membrane potential. Now, what is resting membrane potential? Resting membrane potential is that voltage difference between inside of the cell and then outside of the cell. And that's why we have said this cell, when it is at rest, it is going to be electronegative intracellularly. Okay? Now, the resting membrane potential for the case of this cell is going to be approximately around negative 90 millivolts. Let's first pretend for a minute that it is going to be negative 90 millivolts. Okay, negative 90 millivolts. Now, this cell is at rest and is having too much of electronegativity inside this cell. And we're going to be having a lot of the potassium ions inside this cell, and we shall have a lot of sodium ions which are going to be extracellular. Now, what is going to be happening is that let us now first stimulate this cell. We're going to stimulate this cell, and when we stimulate this cell, we're going to be now having some little bit of what we call entry of cations. Okay, so now let's first stimulate this cell. We are going to be stimulating. We're going to be stimulating this cell. Now, when we stimulate this cell, we're going to be starting to see some cations little bit trying to enter this kind of the cell. Okay, so now you're going to be having entry of what we call cations. Preferably, we're going to be having entry of what we call sodium. Okay, we're going to be having entry of what we call sodium. Now, if we have entry of sodium ions inside this cell, it means the negative polarity of the cell inside is going to be reducing. That means this cell is starting to become electropositive slowly by slowly. Okay, now this entry of sodium ions is going to move the membrane potential from resting membrane potential up to around the threshold potential. Okay, is going to be moving this membrane potential, and then it moves it towards threshold. Okay, so now let's first pretend that this one here is going to be now our threshold potential. And then for the case of this one, let's first pretend that the threshold potential is going to be maybe around negative 70 millivolts. Now, what is very much special is that whenever this membrane potential reaches threshold, we have those kind of the channels that are waiting for the membrane potential to reach threshold. What are these channels? You see these channels here. These channels here are very much special. These channels are what we call voltage gated sodium channels. Okay, these channels here are what we call voltage gated sodium channels. For them, they open only when the membrane potential reaches what we call threshold. Now, what happens is that when this kind of the voltage gated sodium channels, when they open, sodium is going to be starting to rush inside at a very high speed. So now we have now entry of sodium at a very high speed. Now, the too much entry of the sodium ions by the opening of the voltage gated sodium channels is going to be increasing the electropositivity inside this cell. It is going to be moving this kind of the membrane potential. It will move the membrane potential up to around, for example, up to now, for example, this is going to be 0 millivolts. It means that as it reaches 0 millivolts, it means that the whole of inside the cell, it is neither positive nor negative. Okay, it has now washed away all the electronegativity inside this cell. It means there is no positive ions, there is no negative ions. These sodium ions that came in, they were cancelling out the electronegativity. But this end of the sodium ions will further make the inside of this cell to become now electropositive. Okay, so now let's say, for example, up to around positive 10 millivolts. Meaning that at this time, the inside of the cell is now electropositive. Okay, inside the cell now it is electropositive. We have washed away all the electronegativity inside the cell. Now, whenever this cell becomes electropositive, we have washed away the electronegativity, which is naturally inside. We say that this cell is depolarized. Okay, that is now a beautiful term. We say that this cell is now depolarized. Now, when this cell becomes depolarized, we have those kind of the specific type of the channels that are super sensitive to what we call depolarization. Now, what happens is that you see these kind of these channels here, they are sensitive to what we call depolarization. Whenever this cell becomes depolarized, that's when these kind of the channels are going to be opening. What are these channels? These channels here, this one here is what we call voltage gated potassium channel. This channel is going to start opening. Okay, it's going to start opening, and once it opens, you're going to be having now potassium ions starting to leak out. Now, what is the significance of this? Whenever potassium leaves the cell, this cell is going to be starting to get back into its electronegativity slowly by slowly, depending on how fast potassium is going to be effluxing or is going to be moving out. Now, as voltage gated potassium channels are opening, it means now we are trying to pull back the membrane potential back to its what we call its electronegativity. This is what's going to be happening. So now, for a brief moment, you're going to be having a slight kind of the movement back to the membrane potential to around 0 millivolts. Now, what's going to be happening? These voltage gated potassium channels, they are now also going to be telling that also these channels here also slightly a bit open. What are these channels here? These channels here are voltage gated calcium channels. Okay, are voltage gated calcium channels. Now, when these channels open, they are going to be allowing entry of what we call calcium ions. Now, what is very funny at this moment is that the entry of calcium ions and exit of the potassium ions, they are going to be now at this time moving almost at the same rate. What do I mean by this? If there are 2 potassium ions moving out, there are also going to be 2 calcium ions coming in. Meaning that at this point, there is going to be no change in membrane potential. And this one is going to be creating what we call a plateau. Okay, it's going to be creating what we call a plateau. Okay. Now, what's going to be happening now, these calcium channels, these voltage gated calcium channels, they are also going to be opening for a very brief moment, and then they are going to be now closing. Okay, they are now going to be closing, and once they close, we shall have now an extensive amount of voltage gated potassium channels opening. You see these channels here, they are now voltage gated potassium channels, meaning that you're going to be having now an extensive efflux, of what we call potassium. Meaning that if we have an extensive efflux of what we call potassium ions, we are now starting to regain our electronegativity inside the cell. What do I mean by this now? This electropositivity is now also starting to get disappeared. Okay, we are now getting back to our electronegativity, but we have not yet washed all what we call the electropositivity. Now, as more of the potassium keeps leaking out, we are going to be now bringing back our membrane potential. It's going to be coming back to what we call to the resting membrane potential. Okay, so now our membrane potential has now come back finally to what we call to our resting membrane potential, and we said whenever a cell is at rest, it's going to be electronegative intracellularly, meaning that all our electropositivity has now been completely washed away. Okay, we have now regained our electronegativity. Okay, so now whenever we gain back our electronegativity of the cell, we say the cell is repolarized. Okay, meaning that this is going to be a phase of repolarization, and this one here is going to be a phase of depolarization. Okay, it is such another a beautiful thing. So that if you have a wave of depolarization followed by a wave of repolarization, these two waves together is what we call a reaction potential. Okay, if you have what we call depolarization, followed by repolarization. Okay, these two common terminologies together, they form what we call action. They form what we call action potential. Okay, what we call action potential. So now action potential is going to be a wave of depolarization followed by what we call repolarization. Okay. Now, something else which is so much beautiful is that how is this depolarization going to lead into the depolarization of another cell? There's going to be another cell here. And now, during that time when we are having a lot of the cations, before the polarization phase, some of the cations, they start now moving and then they enter into another cell. The question is, they enter into another cell, passing where? We have these beautiful these proteins. These proteins here are what we call the gap junctions. Now, these cations are going to be entering into another cell. Now, if they enter into another cell, if that cell was at rest, for example, on resting membrane potential of around negative 90 millivolts, if we have some of the cations trying to tickle inside there, it means its resting membrane potential is also going to be starting to move towards the threshold. And once reach a threshold, by the entry of these cations from this cell, which is already depolarized, this means that voltage-gated sodium channels are going to be opening. And once they open, a lot of the sodium is going to be rushing in, which will move the membrane potential into what we call around positive 10. And by that time, we say that also the next cell is fully depolarized. And of course, the proceeding processes is going to be the voltage-gated potassium channels, which are depolarization sensitive, are also going to be opening for a brief moment, with also opening of what we call the calcium channels. Okay, this will create this kind of small depression, and of course, we have what we call this kind of the plateau, whereby the calcium ions entering in and the potassium ions leaking out are also going to be on equal interval. And then finally, the voltage-gated calcium channels will close, which allow too much of the potassium ions to be leaking out, bringing back the membrane potential back to the resting membrane potential. So now this kind of the process is going to be happening on the next cell and the next cell and the next cell and the next cell until the entire myocardium gets depolarized. Okay, so now that's how generally how depolarization is going to be coming about. Okay, now that we have seen what is depolarization, what is the repolarization, let's have just a very quick break. Then immediately after the break, we are going to go ahead and see what is a lead, what is electrode, and what is a vector. We meet immediately after the break.